Hello and welcome to Enzymes 3.1. Okay, so there are a lot of different types of enzymes, and each one is specific to a particular reaction. This specificity is the key to understanding efficient functioning of cells and living organisms. So enzymes are protein molecules, which we know that by now. And they really should be defined as biological catalysts. A catalyst is a molecule that speeds up a chemical reaction, but remains unchanged at the end of the reaction. So it's not considered a product or a reactant. Virtually every metabolic reaction which takes place within a living organism is catalyzed by an enzyme. And enzymes are therefore essential for life to exist. So many enzyme names end in ase. For example, amylase. There's lactase. There's so many. So not all, but a lot of times when you see a word that ends in ase, it's an enzyme. So they operate within cells, and often are the ones that do are called intercellular enzymes. Enzymes that are secreted by cells and catalyze reactions outside of cells are called extracellular enzymes. And like all globular proteins, so remember, enzymes are globular proteins, enzyme molecules are coiled into a precise 3D shape with hydrophilic R groups on the outside of the molecule, ensuring that they are soluble. Enzyme molecules also have a very special feature in that they possess an active site. Active sites are regions, usually it's a cleft or a depression, in which another molecule can bind. So this molecule is a substrate of the enzyme. And the shape of the active site allows the substrate to fit perfectly. The idea that the enzyme has a particular shape into which the substrate fits exactly is known as the lock and key hypothesis. The substrate is the key, whose shape fits the lock of the enzyme, and the substrate is held in place by temporary bonds which form between the substrate and some of the R groups in the enzyme's amino acids. This combined structure is termed the enzyme-substrate complex. So if you look at this model, you see the active site has a very particular and specific shape. It has a cleft in its surface, and that is the active site. The substrate molecule has a complementary shape. And in this analogy, the substrate is the key and the enzyme is the lock. So random movement of the enzyme and the substrates bring them together. An enzyme, the substrate complex is temporarily formed. The R groups of the amino acids in the active site interact with the substrate, and it's the interaction of the substrate with the active site which breaks the substrate apart. So an enzyme, which is a product, complex is briefly formed, okay, before the two product molecules leave the active site, leaving the enzyme molecule unchanged and ready to bind with another substrate molecule. An enzyme may catalyze a reaction in which the substrate molecule is split into two or even more. Alternatively, it may catalyze the joining together of two molecules, as when making a dipeptide. The enzyme-product complex is briefly formed before release of the product. It's the interaction between the R groups of the enzyme and the atoms of the substrate which breaks or encourages the formation of bonds in the substrate molecule, forming one, two, or even more products. So with the lock and key hypothesis, this model shows the substrates coming into contact with the enzyme. And here you have let me use my pen here. Okay, here you have the enzyme substrate complex. Okay, here is the enzyme product complex. And then at the end, the enzyme leaves and may be used again. Each type of enzyme will usually act on only one type of substrate molecule, and that's because the shape of the active site will only allow one shape of molecule to fit. The enzyme is said to be specific for this substrate, hence the key and lock model. In 1959, the lock and key hypothesis was modified in the light of evidence that enzyme molecules are more flexible than is suggested by a rigid lock and key model. The modern hypothesis for enzyme action is known as the induced fit hypothesis. Basically, it's the same as the lock and key hypothesis, but it adds the idea that the enzyme, and sometimes the substrate, can change shape slightly as the substrate molecule enters the enzyme in order to ensure a perfect fit. So this makes the catalase even more efficient. And this explains the enzymes that can react with a range of substrates of similar types. When the reaction is complete, the product or products leave the active site. The enzyme, again, is unchanged by the process, so it is now available to receive another substrate molecule. The rate at which substrate molecules can bind to the enzyme's active site, be formed into products, and leave can be very rapid. The enzyme catalase, for example, can bind with hydrogen peroxide molecules and split them into water and oxygen, and release these products at a rate of 10 million molecules per second. As catalysts, enzymes increase the rate at which chemical reactions occur. Most of the reactions which occur in living cells would occur so slowly without enzymes that they would virtually not happen at all. The substrate will not be converted to a product unless it is temporarily given extra energy. Okay, and as you should remember from STEM, this energy is called activation energy. Okay, so increasing the temperature will make molecules move faster. Biological systems are very sensitive to temperature changes. Enzymes can increase the rate of reactions without increasing the temperature. They do this by lowering the activation energy needed for the reaction to occur. They actually create a new reaction pathway or a shortcut. So if you look at this model, okay, this is a typical rate of reaction graph. You have energy here, progress of reaction. So to change a substrate into a product, the energy of the substrate must be briefly raised by an amount known as the activation energy, and this can be done by heating the substrate. When a substrate binds to the active site of an enzyme, the shape of its molecule is slightly changed, and this makes it easier to change the substrate into a product. The activation energy is lowered. So you see here that the activation energy is quite a bit lower using the enzyme. Okay, so then you don't have to heat up. Nothing has to be, the temperature doesn't have to be increased for the reaction to occur. Okay, so you guys should be very familiar with this rate of reaction graph. Okay, here is the activation energy. This is the transition state into which the reaction is about to occur. Here the reaction is occurring, and at the end, here are the products. The products have less energy than the reactants had in the beginning. Therefore, it is an exothermic reaction. Enzyme-controlled reactions proceed 108 to 1,011 times faster than corresponding non-enzymic reactions. As soon as the enzyme and substrate are mixed, bubbles of oxygen are released quickly in the reaction of catalase, catalyzing the breakdown of water. A large volume of oxygen is collected in the first minute of the reaction, which is why you see bubbles. As the reaction continues, however, the rate at which oxygen is released will gradually slow, and the reaction gets slower and slower until it eventually will stop completely. The curve that you see here at its steepest at the beginning of the reaction. The rate of an enzyme-controlled reaction is always fastest at the beginning. And this rate is called the initial rate of reaction. You can measure the initial rate of a reaction by calculating the slope of a tangent to the curve as close to time zero as you can. An easier way of doing this is simply to read off the graph the amount of oxygen given off in the first 30 seconds. So in this case, the rate of reaction production in the first 30 seconds is 2.7 centimeters cubed of oxygen per 30 seconds, or 5.4 centimeters cubed per minute. And remember, on a Cambridge exam, you need to really pay attention whether they're asking for per seconds or per minute. If it doesn't say, you should assume seconds. Okay, that is it for our introductory video of enzymes. We have one more video about factors that affect enzymatic reactions. Thank you.